All right, let's, Miles, if we can jump into polling question number one. We're going to present that, and then I'm going to address another audience question here. But polling question number one, this is for you in the audience. What's your leading rationale for process intensification? You've got a number of options there. I won't read through them, but just make a selection, and we'll revisit those results here shortly. In the meantime, I want to get to a question from Pritam. I think this is a good one, because I started the conversation with the question to Irina and Sanjay. When you looked across the environment of the manufacturing environments in your workplaces, where did you see bottlenecks that could be addressed with process intensification? I think Pritam's question is a little bit more specific in that he's asking, what challenges or bottlenecks did you encounter during the process intensification development? So maybe, Irina, since you're large on my screen, maybe you can tackle that one and give our audience a heads up on some of the challenges that you met when you were addressing the initial challenge.
This is such a great question. I anticipated a lot of challenges from the change. Just the fact that it requires an impactful change to the way we operate. And from an AstraZeneca perspective, where we have a lot of different facilities, different scales, non-GMP and GMP, was just the impactful number of people that run their own facilities in a particular way. We are a conservative industry because we are highly regulated. So the way I manage my stakeholders was to involve them early on. So the number one thing I wanted to prove to myself and my manager, of course, was that we could operate these with no bioburden. So are we testing a particular system in a way that it is closed system for X amount of weeks or a few weeks, X amount of weeks or days? And once we were able to prove that we could adapt the current platform process, the downstream and the upstream, and integrate it, initially welding bags, not connecting to the bioreactor. And we could bring those stakeholders on site for a tour. And it coincided for us during the pandemic, which was a different type of challenge. We absolutely were able to tell them and ask them to challenge us in the technology so we could test accordingly to their expectations. So I think that's the number one, is how do we manage our stakeholders early on, even when we don't have all the answers. So they provide their, I guess, pain points, how they, during the manufacturing, can envision the buffer prep to feed all of these unit operations at once. Or how do we make some sort of turnaround from projects to projects, or roll bags, little things like that, to the more impactful things, which is how are you placing your virus filter versus your UFTF, and how do you run those continuous single passes filters in a way that you don't jeopardize your virus segregation, which a lot of people think about. And I come from, I guess, a line of scientists that is like, do we really need that? And for how long do we still need that if we can prove that our mechanical engineering solutions work for us? So the last, I think the second half of the question was related to, okay, but you might not have capacity internally. How do you go about transferring to an external facility? That's where understanding capacity, demand, and hybrid process development is crucial. The way we develop our platform is to try to not diverge too much from the current mode of operation of our chromatography columns, our filters, where if you were to slice that particular part of the process, the way we run chromatography is exactly the same way we are running in fed batch mode. We just do it in an automated way. So now if we just have a capture integrated with a bioreactor in a CMO of our choice, we can choose to operate that part as intensified and then do the remaining of the process with other unit operations. I'm not saying that's what we're doing. I'm just putting as an option in a scenario out there to also gradually invite CDMOs and CMOs to bring end-to-end continuous manufacturing to their facilities. And often what they do is they start small, but small doesn't mean too much less, because don't forget, those bioreactors often have 7, 10x productivity comparatively to fed batch. So you have a lot of mass to deal with anyway. Everything is just smaller because you can operate upstream and downstream in parallel.
Mm-hmm. Yeah, very good. Thank you. Very thorough answer. Sanjay, what would your response be? Did you just run into any different sort of bottlenecks or challenges as you were implementing some of these projects?
Yeah, I think when we develop a process intensification technologies, we always consider manufacturing aspects into the equation. How are we going to implement this into manufacturing? What's the robustness of the process we're developing, and what's the regulatory impact we could see for that process? And also, when we develop this process and when we tech transfer to external facilities, I think idea is to develop some process intensification technologies which does not require a lot of capital investments.
Mm-hmm.
They're more kind of a consumable part of things. That's where our current goals are, that we could develop this process intensification technologies with no new capital investments.
Yeah.
So it's easy to transfer to external manufacturing or internal manufacturing with the checks and balances in place, what is we required to complete the unit operations.